Another type of DNA damage repair system is the mismatch repair. The mismatch repair system corrects rare base pair mismatches and short insertions or deletions that appear in the DNA after replication. DNA polymerases introduce about one mispaired nucleotide per 10 to 5 nucleotides. So it means if there are 10 raised to the 5 nucleotides, there is chance that one mispaired nucleotide has been introduced in the DNA. However, the 3' to 5' proofreading exonuclease increases replication fidelity by 100 folds by removing these mispaired nucleotides. So even after the proofreading and after removing this mispaired, there remains some mispaired bases. Although an error frequency of one nucleotide in 10 raised to the power 7 may seem extremely low, but it would result in a high mutation rate. The second type of error that occur during replication is the short insertions or deletions. So these short insertions or deletions, they may result from the fact that repeated sequence motifs are present in the DNA, and sometimes these repeated sequence motifs are dissociate from the parent DNA, and then they re-anneal incorrectly to the DNA. So if one motif is removed and it has annealed somewhere else, it cause insertions, and similarly if it has dissociated and but not re-annealed, it may lead to deletions. As a result, the newly synthesized strand will have a different number of repeats than the template strand. Introduction of an insertion or deletion into the newly synthesized DNA is likely to produce a mutation. Cells with non-functional mismatch repair system have a high rate of mutation due to their inability to efficiently repair base pair mismatches, short insertions, or deletions that arise due to replications. Because there are some cells which don't have these mismatch repair systems. So in those cells, the number of such mismatches is more.